The DC-ERG technique can be used for the non-invasive evaluation of retinal pigment epithelium function, to monitor age-related changes or disease progression, and to assess pharmacological intervention effects.
This technique improves upon DC-ERG reproducibility and ease of use by simplifying the capillary electrode preparation. It can also be supplemented with the standalone software application to facilitate analysis.
Begin by carefully sliding a 25-gauge syringe needle through the silicone rubber gasket to the back wall of the electrode holder. Fill the base of the electrode holder with degassed HBSS while slowly retracting the needle, taking care not to introduce bubbles, and replace the threaded cap without tightening. Reinsert the syringe needle to fill the empty space within the cap with HBSS, and hold the capillary horizontally while filling to prevent solution escaping from the other end. Grasping the filled capillary at the bent end, slowly insert the other end through the loosened cap, and tighten the screw cap into place. Tilt the electrode holders with the capillary electrodes facing up to allow any bubbles to flow out, and degas the capillaries in a vacuum chamber for 5 to 10 minutes. Then slowly release the vacuum, and refill the electrode holders and glass capillaries as demonstrated. To make a custom stand for the microelectrode holder, remove the black poly